वेलकम टू एजीपीजी स्टडी। लखनऊ यूनिवर्सिटी के बीएससी एग्रीकल्चर फर्स्ट सेमेस्टर फर्स्ट ईयर की प्रैक्टिकल फाइल फंडामेंटल ऑफ जेनेटिक्स। So let's start. Well, now I start experiment 5. Aim: study of models of DNA and RNA structure. Basic chemical structure of DNA and RNA. Heteropolymers of nucleotides. Monomer composition nucleotide heterocyclic pentose sugar. Phosphate nitrogenous base. RNA polar ribose phosphate backbone. DNA polar deoxyribose phosphate backbone. Nucleotides joined by 3,5 phosphodiester linkages. Nitrogenous bases is side chains. And do you know what is nucleotides? Basically, nucleotides is a genetic material. It is composed of nucleoside and phosphoric acid. And then second, major nitrogenous bases. Nitrogenous bases found in DNA or RNA, purines and pyrimidines. In DNA is AGCT, means adenine, guanine, cytosine, thymine. Now purines. This is the diagram. That includes cytosine, guanine, adenine, uracil. And in DNA is cytosine, guanine, adenine and thymine. And these are the structure of adenine. You have to draw in this. I share you different picture. These are adenine, guanine, thymine, cytosine, and uracil. And B point, how to formation of base, then sugar, phosphate, and then sugar. This is this cycle: thymine, adenine, cytosine, guanine. In this, DNA and RNA are rich composed of 4 different nucleotides which differ in their nitrogenous bases are the same in DNA, RNA. Adenine, guanine, and cytosine, the 4 bases in DNA is thymine. DNA is thymine, and in RNA it is uracil. To study about the test cross and back cross. This is tall capital T, double capital T, and double small t. This is dwarf recessive, and then we get capital T F1 generations. Back cross: when the F1 progeny cross with any one of its parents is called back cross. Back cross can be dominant or recessive back cross. Dominant back cross: in these cases, F1 individual is crossed with dominant parent is called dominant back cross. In these cases, all progeny shows dominant character like this. In this, all progeny are tall. And in test cross, in this case, F1 individual is crossed with recessive parents and the progeny shows 50% tall and 50% dwarf. This is also known as recessive back cross. You can see in this 50% is tall, 50% is dwarf. Then in 7th experiment, sex-linked inheritance in Drosophila refers to the pattern of inheritance shown by genes that are located on either of the sex chromosomes. Genes carried by X chromosome referred to as X-linked genes. Genes carried by Y chromosome referred to as Y-linked as choleric genes. Example: eye color is Drosophila is carried by X chromosomes. 4 cross, monohybrid cross, self-linked, test cross, and reciprocal cross are generally done to study inheritance pattern in a population. Then monohybrid cross. In monohybrid cross, red eye female WW cross with white eyed male. capital W. All dominant parent type red eye were produced. Female produce, however, all carriers. Note, red eye is dominant over white eye and the gene is carried by X chromosome. This is monohybrid cross. Red eye carrier female cross with red eye male. So here we get 75% dominant type and the 25% recessive type, white eye, were produced with ratio. And in second point, all females are red eye, while 50% males are red eye and 50% males are white eye. Test cross. Red eye 50% female red eye and 50% female are white eye. Red eye females are carrier. 50% males are red eye and 50% males are white eye. Test cross ratio is 1:1. Reciprocal cross. इसमें क्या होता है वाइड आई फीमेल क्रॉस विद रेड आई मेल. Here we get all females are red eye and all males are white eye. You can see in this selfing and test cross, then reciprocal cross. First point is इसमें जो एक्स क्रोमोसोम जो भी जीन कैरी करता है वो ओनली एक्स लिंक्ड रिसेसिव ट्रेट को ही डिटरमाइन करता है एंड ओनली मेल पॉपुलेशन को मैनिफेस्ट करता है. एंड इन सेकेंड पॉइंट, in case of autosomal inheritance, males and females are equally affected. इसमें दोनों affected होते हैं and as observed in autosomal process we made. And in third, while in X-linked inheritance, numbers of males are more affected in than males. Since the gene for white eye, red eye are present on X chromosomes. Note, in all four processes, with 4 males are white-eyed, monohybrid cross none, selfing 1 male, test cross 1 male and 1 female, and reciprocal cross 2 males are generally done to study the inheritance pattern in a population. Experiments on epistatic interactions including test cross and back cross in a category of individual are result of expression of genes. कुछ कैरेक्टर गवर्न करते हैं पर्टिकुलर जीन्स एक्शन को, पर्टिकुलर जीन एक्शन के द्वारा कुछ गवर्न होते हैं, whereas there are certain characters that are expressed with effect of some dependent genes. There is some phenomenon in genetics that is used to study the effect of genes on the expression of other gene or genes. Epistatus. Epistatus ये टर्म बैटसन ने 1909 में दिया था, जो डिस्क्राइब करता है द इफेक्ट ऑफ पर्टिकुलर जेनेटिक वेरिएंट मार्क्स बाय ऑथर लोकस. इसमें क्या होता है, वन एक पर्टिकुलर जीन होता है वो डिपेंड होता है वन और मोर मॉडिफायर जीन्स पे, उसको एपिस्टेटिस बोलते हैं. इन एपिस्टेटिस इफेक्ट ऑफ अ जीन मे बी मार्क्स बाय अदर जीन और जीन्स. द जीन विच अफेक्ट्स और मार्क्स अदर जीन, ऐसे जीन जो इफेक्ट करते हैं और अदर जीन को मार्क्स करते हैं उनको एपिस्टेटिक जीन बोलते हैं. एंड द मार्क जीन इज कॉल्ड hypostatic gene. The segregation of genes occurs in F2 generation of the individuals. Static interaction. The statistical description of an expression of statics is termed interaction. It expresses the interaction between genetic factors at different locus that cause epistatic for a particular character. Among the various types of epistatic interactions, there are mainly 6 types of epistatic gene interaction are observed. These are and recessive epistatics. The ratio is 9:3:4. In this case, इसमें डोमिनेंट और रेसेसिव एलिलीज को मार्क्स करती हैं एट द अनदर लोकस पे. ठीक, इसमें एक्सप्रेशन और डोमिनेंट और रेसेसिव एलिलीज को ही मार्क्स करती हैं डिफरेंट अनदर लोकस पे. इसी को द इंट्रैक्शन इज ऑल्सो कॉल्ड एज सप्लीमेंट्री एपिस्टेटिक्स. एग्जाम्पल, Grain color is maize, maize heaps, three colors, three colors, purple, red, and white of grains. Presence of two dominant genes P and R, that is PP and RR, result into purple color. Presence of dominant allele R and recessive P, that is small p and capital R, result into red color, whereas homozygous recessive alleles small p and small r express white color in maize. Now, if a cross is attempted between capital P and capital R purple grain and small p and smaller white grain, then the plants is F1 will be purple. तो जो F1 के जो प्लांट्स होंगे वो पर्पल होंगे, whereas F2 में क्या होगा, segregation ratio of purple and ratio in white will be 9:3:4, इस टाइप से. The small r allele is recessive and show epistatic effect under the homozygous presence, and the plants in F2 with capital P and capital R will be purple grains, and small p and capital R will have red colored grains, and white color of grains will appear under small r condition. Thank you for watching my video. I hope आपको मेरा वीडियो अच्छा लगा होगा. Keep watching and please like, share and subscribe to my channel.